Hi, welcome to the Physionic podcast. My name is Nicholas Verhoeven. I'm a PhD student in molecular medicine. I have my master's in exercise physiology. And, well, that's all the introduction that I will be giving. Today, we're going to be discussing three different topics. The first one is on pregnancy and genetic changes that occur in preterm children and how that relates to full-term children. So some new research in that regard. The next one is going to be on printing skin, which is a really cool biotechnology, bioengineering feat that it's actually been around for a few years, but now they've really, I wouldn't say perfected it, but they've made it much more accessible for a lot of individuals. So I'll be going into what that exactly means. And the final topic is going to be talking a little bit off the cuff on something that was brought to my attention by a comment under one of the videos. And I thought I'd address it because I think it's pretty important, and that is related to why do scientists study such obvious phenomenon? Like things that you observably already know happen, so why do we need data to back it up? So I'll cover that, and there's definitely some nuance there that I think a lot of people don't really consider because they're not necessarily in the scientific field. So, as usual, I'll have timestamps for you so you can skip to whatever topic you'd like to discuss. And with that said, let's jump into the first topic. So the first topic is talking about pregnancy and genetic changes. I've got it labeled or titled as longer pregnancy and genetic changes. Now, this research comes out of the esteemed Karolinska Institute, which is incredibly famous institute or university. I guess, I don't know if it's a university. I think it is, but it's definitely a research institute where a lot of famous researchers are located, and as well as many have studied for short periods of time there to learn techniques and all kinds of different things. It's really famous for exercise physiology, but it's actually famous for a lot of other things as well. So essentially what this research shows is that with the length of pregnancy, there is an impact on our genes as children. And it's not just as children. It turns out that the longer the pregnancy, the different genetics we have. And I'm not saying the base genetics, like the actual base genes themselves are the same. However, there are other things that can change actually after we're born. So our DNA is pretty much set in stone, other than, maybe mutations and things like that, which make some small alterations, but usually our body ends up fixing those. So our genetics are essentially set. I mean, you get some genes from your father and you get some genes from your mother, traditionally speaking. So those are set. However, there's something called epigenetics, which is where you have these tags that are put on particular genes to either tell molecules and proteins and enzymes and all these different things to either attach to those genes and express those genes, or to not attach those genes and knock them down, or to attach to those, to not attach to those genes. I just confused myself. They can either attach to the genes and knock it down, or they can be dissuaded essentially from attaching to that gene and expressing it. So the reason for that is because when you have these epigenetic changes, and that's through things like acetylation, methylation, typically when you're talking about the actual gene sequence itself, you're talking about methylation. However, DNA are wrapped around, they're not just in a helical pattern, which is the traditional view of DNA, but they can actually be wrapped around these protein blocks known as histones. And when they're wrapped around those histones can be either tightly wound or they can be loosely wound. So imagine, having a block of wood and having a string tied all the way around it. So it's looped all the way around it. And you have two people, or maybe even just one person, that's pulling on both ends of that string and really creating some torque between the string and the histone, or the block in this situation. it's more difficult to loosen that string and actually get to, let's say, the underside of that string, as opposed to if you don't have that person pulling on it and the string is very loose on that block of wood. Then it's really easy to just pick up the string and just kind of turn it over. So it's a similar process with DNA, right? So if you have acetylation of the histones, and this is certainly variable, sometimes acetylation leads to the tightening of the histone or the DNA around the histones, and sometimes it leads to the loosening. So this acetylation is a modification known as an epigenetic modification. And the same thing is with a methylation. And you don't have to get too bogged down with what is acetylation or what is methylation. Just know that they're tags. They're tags that can be attached to DNA or to substances and proteins and structures, complexes that are associated with DNAs that either allow DNA to be read or to not be read, and promote its reading or knock down its reading. That's the point that I'm trying to get across here. So that was a big backstory to explain to you what this actually means. So I told you that the genes themselves, the DNA themselves, do not change. So that seems to be consistent for all the children. However, the epigenetic modifications, meaning that those tags, which are actually reversible, so they do change based on particular circumstances. If you're exercising, if you're eating certain foods, if you're at a certain temperature, like literally everything in our environment affects our DNA and how our cells end up closing DNA or opening DNA and expressing DNA. I mean, all kinds of different things. So what they found with this research is that children that were preterm had differences in their epigenetic modification. So they had tags in different places than the children that ended up going full term. And then they took it a step further. And while a lot of those epigenetic changes ended up reverting back to the normal term level, so the preterm babies, over time, most of their epigenetic changes ended up changing back to the full-term babies comparison, 17% remained altered for years. And when I mean years, I'm talking all the way to adolescence. And I think that's as far as they tracked it. I'm not sure if there's a potential there that it might maintain for longer than just adolescence. So in so much that essentially these quote-unquote temporary tags are rather permanent. I mean, technically you can still change them, but 17% lasting till adolescence. I mean, you're talking, 12, 14, 16 years here of these tags that are differential compared to full-term babies. So that is a really interesting line of research, because what it could mean, and they're not saying that this is necessarily the case, but they are saying that there's a difference here. That's what we know. That's what the research shows. There's a difference. Now, what else do we know about preterm babies? We know that they tend to have higher incidence of different pathologies, maybe not quite the same level of brain development or body development of some sort. There's all kinds of different things. You'd have to talk to a specialist, or, once I delve into that research, I can speak more on it. But for the time being, all I know is that there are pathologies associated with preterm babies. Sometimes preterm babies turn out perfectly fine, but the incidence is much higher for a preterm baby, based on my limited knowledge of what I know so far. But then that means that if that is the case, if there's an increase in pathology in those preterm babies, then maybe some of that could be attributed to the fact that there's 17% of the DNA changes, the epigenetic changes, are different between the preterm versus the full-term babies. Now, in one respect, that's kind of exciting, or it's kind of scary, but on the other hand, it's also kind of exciting, because I've mentioned this a few times now, epigenetic changes are typically temporary. Now, we have difficulty making or changing the epigenetic changes on an isolated case, on that gene. Like, this is the gene that we want to remove the tag or add a tag or whatever it might be. So in that way, it's discouraging. But in an encouraging way, it's really cool because we can actually use different drugs to lead to less or more of this tagging, general tagging that's occurring across the entire DNA sequence. So maybe if we can figure out ways of targeting some of that tagging, some of those drug effects to particular genes, we might be able to rescue some of those effects and may lead to better outcomes for preterm children. But for now, all we know is that there are differences, and if those differences are associated, we don't know that yet. But I would imagine, if I had to throw my hat in the ring, I would imagine that there is an association between the pathologies and these epigenetic changes. All right, so moving on to topic number 2, which is printing skin. I've got it labeled as printing skin. So this isn't my area of expertise. I'm not a bioengineer. I'm not a biotechnologist. However, I can speak on some of the biological aspects of what this actually means. So researchers out of the University of Toronto have figured out a way to create a portable, and that's kind of the big thing because they actually created this a few years ago, I think in like 2018. So it's been around for a few years, but this one is actually portable because they've made a lot of different iterations of this printer, and well, which is implied by the title, printing skin. They're literally taking burn victims, and we're talking people that have from minor burns all the way to extreme burns all across their body where they've lost most of their skin. And what they can do is they can print new skin, but not exactly. So what it is it's called a bio-ink. And that bio-ink is essentially a paper-like filament or structure that can be printed over the holes in the person's body. I mean, essentially these gaping areas of skinless area, which is really dangerous because you're extremely prone to infection and extremely prone to all kinds of other issues. So they can not only cover that area, but within 2 minutes, so less than 2 minutes, this bio-ink starts to kind of ingrain itself into that open area. And now what makes that possible? So what makes that possible is that in that bio-ink, in that filament, that paper-like filament, there are what are called mesenchymal stem cells. So mesenchymal stem cells are a slightly limited version of a regular stem cell. I mean, they're stem cells for sure, because they can turn into a series of different tissues. They can turn into muscle tissue. They can turn or muscle cells. They can turn into adipose tissue, fat tissue. They can turn into bone tissue. They can turn into, well, obviously, skin tissue. So the idea is that if we place these mesenchymal cells in a layer, that they will then gravitate towards those open areas and then get the signal from the body to turn into, the process is called differentiate into skin cells. And that's what they end up finding. So it allows for a much faster healing process. And what's really cool about this is that it's different from a skin graft where you just take skin from a healthy individual or a different part of the body and just place it there, because you're actually getting new mesenchymal cells that are actually integrating into that skin and creating a whole new layer of skin. So it's much faster. And I think that they added something, I forgot what exactly they did, but they changed one formula to make it far more effective. So they added something to the mesenchymal stem cells to make it easier for them to migrate or for them to end up integrating into the skin layer and whatnot. But I thought that was so cool that now hopefully we'll be able to have these situations where individuals can come in from an extreme burn and they may not die because we'll have skin that's essentially able to kind of produce itself. Now, the only drawback from this, I imagine, is that this is going to be insanely expensive, just insanely expensive. I mean, one, the technology itself, two, the fact that it starts to happen, I should say, in less than 2 minutes. I'm not saying that it's like you just have skin 2 minutes later. It starts to happen after 2 minutes. But three, and probably the most important or the most expensive, is that where are you going to get these mesenchymal stem cells? I mean, they don't grow on trees. Well, I guess if you were to talk to a plant biologist, maybe I would imagine stem cells do. I don't know. Now I think about it, I am not a plant biologist, so I could not tell you. I imagine that plants have stem cells, though. So maybe they do grow on trees. I take it back. However, they won't work for humans. I know that much. They won't work for humans. Or maybe I guess I shouldn't say I know that much. Maybe in 50 years we'll have research of we're using plant stem cells and we're turning them into human stem cells. Who knows? That would be incredible. Oh, wow. I feel bad for Mother Earth because we're gonna be chopping everything off of it and just extracting as many cells from it as possible. Anyway, no, I guess it couldn't be because plant cells are not eukaryotic, so that would not work. Boom, there you go. Thrown it back to some biology, some basic biology. So mesenchymal stem cells, you need to get that from somewhere. And then, of course, while they are certainly more malleable and they're not going to be as aggressively attacked by the immune system or whatever it might be, it would be certainly tricky to, quote unquote, convince those cells to start differentiating into skin cells. And where are you going to get them? And wherever you get them, that's going to be an involved process to separate out mesenchymal stem cells and have them create this bioink just for your body. And for especially if, like, let's say you've got 70% burns, I mean, that's a ton of material. That's a ton of cells that they're going to have to use. So I imagine this is going to cost so much money. But hopefully over time, as we get better and better and better at this stuff and we get more innovative with this stuff, that things will slowly start to reduce in price. Okay, so that was topic number 2. And now, topic number 3 is quickly addressing a comment that I received, not in bad spirits or anything, but, it got me thinking, and I was like, well, I mean, I think it'd be interesting, I think it'd be informative to actually cover this, because I get that people sometimes don't understand how the scientific process works in certain regards. So, what is the issue? So, somebody commented under a recent piece of content that I released that was looking at muscle size, the association between muscle size and muscle strength. And the association was quite strong. It was really strong, actually, that with an increase in muscle size, you see an association with an increase in muscle strength. Okay, I get it. That seems really obvious. But, I mean, granted, for me, I feel much better having actual scientific evidence to show that. And granted, in this case, we are talking about an association, so who knows? But it at least gives some line of thought towards, or some credence to the thought that, yeah, I mean, the larger your muscles are, the stronger you'll be. Now, I mentioned in that content that is not necessarily going to be 100% responsible for the gain in strength. Like, I'm sure you know of individuals that are pretty small, they may be lean, or they might just be skinny, but they're really, really strong. Well, those individuals would fall into some other reasons as to why they gain strength. And it's not necessarily due to their muscle size. But of course, granted, most likely, if they did increase their muscle size, they would get even stronger. So that is something to consider. Now, the big gripe was, well, why are we studying this? I mean, this seems so obvious. Why are we wasting resources? And that was kind of the big point here. Why are we wasting resources to figure out something that is obvious? Like, you see a person getting bigger and bigger and bigger, and by default, they're getting stronger and stronger. Now, my first argument against that is that while that does seem to be a waste of money, it is nice to actually have science to back up claims, regardless of how obvious it seems on onset, just by looking like, oh, well, I've been to the gym for 5 years, and throughout those 5 years, I've seen that, if a person goes to the gym consistently and they get bigger and bigger and bigger, that they get stronger and stronger and stronger. They can lift more and more weight. Now, okay, great, that's fine, but it's still anecdote. So why not study that and actually get some data points and actually use a statistical method to actually figure out if that's the case? Now, most likely it will be the case, and that's what we found, at least with this association. However, the second argument, I think, is even stronger than the first argument. And I like the first argument just because I like to have data. I like to have actual statistics that I can pore over and figure out if I agree based on the statistical model that they used or don't agree. And having a series of different studies to cover that is really important as well. I'm always going to say that. A single study is great. It's informative and it's helpful, but a whole literature base saying the same thing or roughly the same thing to varying degrees, that is far, far more powerful than a single study, no matter how great that study was designed. So then why would we design a study that would look at something so obvious? Well, the answer is we don't. That's the thing. So the study that I was covering for that content was actually not looking at that at all. They just happened to have extra whatever, like muscle. So in this situation, they were looking at muscle cells, the size of the muscle cell, so the increase in muscle cell size and squat strength. So they had the information on muscle cell size from their actual hypothesis, from their actual study. The actual study was looking at creatine and whey protein, and yes, that content will be coming out shortly, but looking at creatine and whey protein and how it impacts muscle strength as well as muscle size. So that's what they really wanted to figure out. And of course, you could use the exact same argument, well, then why are we studying that? Don't we have enough studies on creatine? Well, we had to start somewhere. We had to figure out somewhere. And there's certain nuances that they ended up changing to make this study a little more, to be informative. Because when you write a grant, you have to have something that makes your study innovative. You have to have a significant reason for why you're studying this. You can't just change one little variable and just say, okay, well, we changed this little variable, give us, $500,000 to study this. That's not how things work. You have to have several different things that are novel about your study, or you're going to be covering a series of different subjects all within this one study. And that's what happened in this study. So while they were looking at creatine and they were looking at whey protein, they also thought, well, we already have this information about squat strength because we studied it with the creatine and whey protein, and we already have the muscle size, the cell size differences from the creatine and whey protein study. So why don't we just make a graph? That's literally all they had to do. They didn't study it. They weren't going out there to actually study that. They just were like, well, we have this information. Let's just publish it. Let's just throw it in there. Why not? It's informative. It's helpful. It's interesting. I mean, at the very least, it's interesting. So we'll just throw it out there. And they made a correlation based off that. They had to do no extra work. They probably just put in their data into a program like GraphPad or something like that, which is extremely popular. It's what I use. And then it just made a graph. And they were like, okay, good, done. We'll throw it into the paper. So in terms of resources, they probably spent no extra money, no extra time on any of this. I mean, in terms of extra time, maybe half a day between the writing and the creation of the graph. The creation of the graph probably took 30 minutes, and the writing probably took a few hours. That was it. Yeah, don't think that just because we have information that necessarily, it always relates to the title. Sometimes you can, and not sometimes, often you can gain information about a topic, even though really just in the body of the paper, because there's several different figures. There are many different experiments that are done throughout a study, and that's what allows us to glean some information that might be independent of the initial hypothesis or the initial study topic that they're covering. In this case, whey protein and creatine. So, yeah, essentially all I'm trying to say is when, especially, I think to speak on that a little bit more, I also think that when people talk to an expert in a field, they might ask, well, has there been any research on Well, let's take the exact same example, a little bit different. So this was actually done in the study as well, looking at contractile proteins like myosin and actin, which are these filaments that essentially crawl across one another to shorten a muscle cell or to lengthen the muscle cell, just like in an eccentric or concentric contraction. So they looked at contractile protein amount and squat strength. So if a person says, well, has anybody ever looked at contractile protein amount, so the amount of protein that's within a muscle cell and how that relates to strength? And while there might be studies that specifically look at that, often the expert will have to dig into their memory bank or into their research library that they've come up with over time and say, oh, well, actually, so there's no study that specifically says in the title, we are looking at contractile protein versus squat strength. But because maybe it was covered in some sub-aim, in some subcategory of a paper looking at, I don't know, let's say bench press strength, squat strength, and deadlift strength in 35-year-olds and 70-year-olds, okay? So now you have two different populations, you have three different exercises, and you're trying to figure out, okay, well, what are some of the differences between 35-year-olds and 70-year-olds? So now you're getting some clinical data on a little bit elderly individuals versus younger individuals and seeing what are some of the differences there. So, but by studying that, you can isolate some of that information just down to the 35-year-olds and figure out, okay, well, we also have information on contractile protein, so why don't we just add that into the supplementary information just so in case somebody's interested in that, they can also look at that. So it doesn't always come down to the title, and that's another reason why you don't want to necessarily just read an abstract. Beyond the fact that the conclusions of the abstract may completely be wrong, so you need to be able to actually read and decipher the literature for yourself. But that's certainly a topic that a lot of people hammer home, which is absolutely correct. However, again, the added benefit of that, just like I'm mentioning, is that there are hidden gems of information that you can glean from a study that is originally not looking at that topic, or they're looking at it from a very loose perspective. They're looking at something completely different, and they just happen to also have that data, and they happen to report that data, and you are then able to extrapolate information from that data in a way that helps you with your topic that is not necessarily related to that study or the main topic of that study. So hopefully I'm getting that information across. So that's essentially it. I mean, that, I just wanted to make those two arguments so people are aware that people aren't just, researchers are intelligent. We're not idiots. And we try and do several things at once. We're not just going to do one thing. Okay, well, if I have these cells and if I add this one compound, if I add curcumin extract to the plate and wait 1 hour, I'm just going to measure this one thing and I'm just going to have a paper out of it. I mean, that's not going to get published. Not published by any journal that matters. That's big. That's high impact. You would have to do many experiments. So let's say you did that, right? You added the curcumin. Okay, you find that instead of measuring one effect, you decide I'm going to measure three effects. I'm going to measure how it impacts glucose uptake. I'm going to measure how it impacts gene regulation or gene expression for FOXO, which is a protein or a molecule that's involved in glucose regulation, a whole bunch of different things, growth, all kinds of things. And I'm going to measure mTOR activity. So those are three different things that you're measuring. So now you're starting to broaden things out. And then let's say that, okay, so you found that information out. Now you want to find out, okay, well, what if I add a protein synthesis inhibitor as well as this curcumin extract? Does it have some sort of impact? So does curcumin extract have some sort of a helpful impact on increasing protein synthesis or blunting the effect of the inhibitor or whatever it might be? And then you start getting more information and you start piecing all this puzzle together, and it gives you several puzzle pieces as opposed to just one isolated puzzle piece to a board of a puzzle that you don't know exists. Like, that doesn't help. Just holding a puzzle piece in a dark room makes no difference whatsoever if you can't see the actual board in front of you. So being able to have several puzzle pieces and having a light there where you can actually see what the puzzle is supposed to look like or what it's going to start looking like, that gives you a lot more information. And each one of those puzzle pieces can be really important and really informative in certain ways and in certain contexts. So yeah, researchers aren't idiots. We're not trying to waste people's money. I'm not going to speak for everyone, of course. We do stupid things, no doubt, but that is part of the whole learning process. So yeah, that's my point. Not really a rant, but I wanted to cover it so that people feel a little more educated on the whole process when it comes to reading papers and how to interpret papers. And I'll certainly be covering more topics on this as well. Okay, so that's essentially where I'm going to end things. I will say I just got news that Wake County, which is where I grew up for most of my life, is the only county in North Carolina that's been infected with coronavirus. I just got that news, so I just had to tell my parents and tell all my friends in North Carolina how unfortunate it is for us. I'm not in North Carolina right now, but that is incredibly unfortunate that the one county that most of my friends are in North Carolina now has coronavirus. Great news, being completely sarcastic. Anyway, with that said, stay safe, stay safe from coronavirus, and I hope that this was informative. Hopefully you got something out of it, and I wish you a wonderful, wonderful day, and I can't wait to speak with you next time. Have a good one, guys. See ya.